Welcome everyone, this is Education Lesson. We're continuing our lecture on Intro to Physiology. Today we'll be covering Module 7, Blood, Respiration, and Host Defense. These three systems can seem separate, but they share one job: keeping every cell in your body supplied with oxygen and protected from harm. Over the next 20 minutes, we'll follow a single thread: the red blood cell and oxygen it carries. From the blood itself, through the immune cells that ride alongside it, all the way to the lungs where that oxygen is loaded. Let's get started. Here's what you should be able to do by the end of this lecture. First, describe what blood is made of and what each part does. Second, walk through how bleeding is stopped and how blood types are determined. Third, tell the difference between our two arms of immunity and describe inflammation. Then we shift to breathing. Explain the mechanisms of a breath, and read a spirogram. Analyze how gases cross into the blood in the lungs, and finally interpret the oxygen-hemoglobin curve and describe how carbon dioxide is carried and how your brain controls each breath. Notice these 6 goals map neatly onto our 3 units: 2 on blood, 1 on immunity, 3 on respiration. Keep them in mind as checkpoints. Here's our roadmap: 3 systems. 1 shared job. 1 unit. Unit 1 is blood. Its composition, the red and white cells, clotting, and blood typing. Unit 2 is host defense, innate immunity, adaptive immunity, and the inflammatory response that links them. And Unit 3 is respiration, the airways, the mechanics of breathing, lung volume, gas exchange, and the control of it all. Blood and respiration are tightly coupled by oxygen, and immunity travels in that same fluid. So everything here connects. Let's start with blood itself. Think of blood as a liquid connective tissue, about 5 liters in an adult. It transports gases, nutrients, waste, hormones, and heat, and it carries the cells and proteins of your defenses. Everything downstream depends on it. If you spin a tube of blood in a centrifuge, it separates by density into 3 layers. On top, just over half the volume, about 55%, is plasma, the straw-colored fluid. At the very bottom, around 45%, are the red blood cells. That percentage has a name: the hemocrit. And squeezed in between is a thin layer, less than 1%, called the buffy coat. That's your white blood cells and platelets. Together, the red cells, white cells, and platelets are the formed elements. One thing worth flagging now: only the white cells are complete cells. Red blood cells and platelets are really specialized fragments, and that shapes everything they can and can't do. Let's look closer at plasma. It's about 92% water, and that water is doing real work. It's a solvent that dissolves everything, and a heat sink that spreads warmth around the body. Dissolved in it are electrolytes, like sodium and bicarbonate, nutrients, wastes, gases, and hormones. But the part to remember is the plasma proteins, about 7%. 3 groups: albumin, the most abundant, which holds water in the bloodstream by maintaining osmotic pressure; globulins, which include your antibodies and various transport proteins; and fibrogen, the raw material for blood clots. One quick vocabulary note: if you let blood clot and remove those clotting proteins, what's left is called serum. Now the red blood cell, a cell built for one job: carrying oxygen, and stripped of everything that doesn't serve it. It's shaped like a biconcave disc, which maximizes surface area for gas exchange and lets it bend through the narrowest capillaries. It has no nucleus and no mitochondria. That interior is packed with hemoglobin instead. And because it has no mitochondria, it doesn't burn any of the oxygen it's carrying. Each hemoglobin molecule has 4 iron-containing heme groups, so it binds 4 oxygen molecules. That's why over 98% of oxygen in your blood is bound to hemoglobin rather than dissolved in plasma. Red cells live about 100 in 20 days, then get recycled in the spleen and liver, with the iron salvaged for reuse. So how does the body keep red cell numbers right? Through a classic negative feedback loop. The trigger is low blood oxygen, from blood loss, high altitude, or lung disease. When the kidneys sense that hypoxia, they release a hormone called erythropoietin, or EPO, into the bloodstream. EPO travels to the red bone marrow and ramps up red cell production over the next several days. More red cell means more oxygen delivered to the tissues. And once oxygen is restored, the signal shuts off. That's the negative feedback closing the loop. One caveat: this only works if the raw materials are present: iron, vitamin B12, and folate. Run short on any one and you get anemia. Now the white blood cells, the leukocytes. There are 5 types, and a handy mnemonic for their frequency is never let monkeys eat bananas. Neutrophils, lymphocytes, monocytes, eosinophils, basophils. We group them 2 ways. Granulocytes, neutrophils, eosinophils, and basophils, have visible granules. Neutrophils are the most common and the first responder that eat bacteria. Eosinophils target parasites. Basophils release histamine and inflammation. The granulocytes are lymphocytes, the B and T cells behind adaptive immunity, and monocytes, which become macrophages. Here's the key idea. White cells don't do their real work in the blood. They slip out of the capillaries by a process called diapedesis and patrol the tissues. When a vessel is injured, hemostasis stops the bleeding in 3 overlapping phases. Phase 1, vascular spasm. The vessel constricts immediately to slow blood flow. Phase 2, the platelet plug. Platelets stick to exposed collagen at the wound, activate, and recruit more platelets into a temporary plug. Phase 3, coagulation. A cascade of clotting factors converts the soluble protein fibrogen into fibrin, a mesh of threads that traps cells and forms a stable clot. That cascade runs through 2 pathways, intrinsic and extrinsic, that converge to make thromboin, which makes fibrin. And once the vessel has healed, a process called fibrinolysis uses the enzyme plasmin to dissolve the clot and clear it away. Blood typing comes down to the antigens, molecules on the surface of red blood cells. In the ABO system, type A has the A antigen, type B has the B antigen, type AB has both, and type O has neither. Here's the twist. Your plasma carries antibodies against whichever antigens you lack. So type A blood has anti-B antibodies. And if it meets type B cells, it attacks them. That's why type O negative is the universal donor. No antigens to trigger a reaction. And type AB positive is the universal recipient, with no antibodies to attack. The Rh factor adds the plus or minus. It's whether you carry the D antigen. Anti-Rh antibodies only form after exposure, which is why Rh matters so much in transfusions and in pregnancy. That covers blood. Now let's turn to how the body defends itself. Immunity has 2 arms that work together: a fast, general, innate system and a slower, highly specific adaptive system that remembers. And bridging them is the inflammatory response. Let's contrast the 2 arms. Innate immunity is nonspecific, and you're born with it. It responds within minutes and treats every threat the same way. It includes your physical and chemical barriers: skin, mucus, stomach acid, plus phagocytes and natural killer cells, and responses like inflammation, fever, interferons, and complement. Crucially, it has no memory. Adaptive immunity is the opposite. It's specific, it's acquired over time, and it takes days to ramp up. It has two branches: humoral immunity, where B cells make antibodies, and cellular immunity, where T cells destroy infected cells. And its signature feature is memory. After the first exposure, a second encounter with the same antigen triggers a faster, stronger response. That's exactly how vaccines work. Inflammation is the innate system's local response to injury, and you can recognize it by 4 cardinal signs: redness, heat, swelling, and pain. All 4 trace back to 2 changes in the local blood vessels. Redness and heat come from vasodilation, more warm blood flowing to the site. Swelling comes from capillaries being leaky and letting fluid into the tissue. And pain comes from chemical mediators and pressure stimulating nerve endings. The sequence goes like this. Injury triggers the release of chemicals like histamine and prostaglandins. Those cause vasodilation and increased permeability. Phagocytes are recruited to the area, squeezing out of the vessels and following chemical trails. And finally, they clean up debris and the tissue repairs. It's uncomfortable, but every step has a purpose. Now to our largest unit, respiration. Keep the oxygen thread in mind. We've seen how blood carries oxygen and how it's defended. Now we'll see where that oxygen actually enters the body. The lungs, and how breathing is powered and controlled. Let's divide the respiratory tract by functions in 2 zones. The conducting zone is the plumbing: nose, pharynx, larynx, trachea, bronchia, and bronchioles. It moves air and conditions it, warming, humidifying, and filtering. But no gas exchange happens here. It's called anatomical dead space. The respiratory zone is where the action is. The respiratory bronchioles, alveolar ducts, and about 300 million alveoli, tiny sacs where gas actually crosses into the blood. That exchange happens across the respiratory membrane, which is astonishingly thin: the alveolar wall, the capillary wall, and their fused basement membranes, all told about 1 cell thick. Thin enough that gases cross in a fraction of a second. How does air actually move? It comes down to Boyle's law. At a fixed temperature, pressure and volume are inversely related. Change the volume of the chest and the pressure changes, and air flows to even it out. On inspiration, which is active, your diaphragm and external intercostal muscles contract. The chest expands. The pressure inside the lungs drops below atmospheric, and air flows in. Expiration, at rest, is passive. The muscles simply relax. Elastic recoil shrinks the chest. Pressure rises, and air flows out. 2 properties matter here. Compliance is how easily the lungs stretch. Fibrosis lowers it. Emphysema raises it abnormally. And surfactant is a substance that lowers surface tension inside the alveoli, keeping those little sacs from collapsing every time you exhale. This is a spirogram, a recording of the air you move over time, and it lets us define lung volumes and capacities. Quiet, normal breathing moves the tidal volume, about half a liter. On top of that, you can force in a lot more, the inspiratory reserve volume, around 3 liters, and force out extra below a normal breath, the expiratory reserve volume, about 1 liter. And there's air you can never exhale, the residual volume, roughly 1.2 liters, which keeps the alveoli from collapsing. Capacities are just sums of volumes. Vital capacity, everything you can move in one big breath, is about 4.6 liters. Add the residual volume and you get a total lung capacity, around 6 liters. Note that residual volume can't be measured by spirometry, since you can't blow it out. Gas exchange runs entirely on partial pressure gradients. Each gas diffuses from where its pressure is high to where it is low. Look at the numbers in milliliters of mercury. In the alveolus, oxygen sits at about 104 and carbon dioxide at 40. The venous blood arriving is the mirror image. Oxygen down at 40, carbon dioxide up at 45. So oxygen diffuses from alveolus into blood, and carbon dioxide diffuses from blood into alveolus, until they nearly equalize, almost instantly. Now, for this to work efficiently, airflow and blood flow have to match, region by region. That's ventilation, perfusion matching. The lung tunes it locally, where oxygen is low. Arterioles constrict to send blood elsewhere. Where carbon dioxide is high, bronchioles dilate to bring in more air. This is one of the most important graphs in physiology, the oxygen-hemoglobin dissociation curve. It plots how saturated hemoglobin is against the oxygen pressure around it, and its S shape is the whole story. The plateau at the top means that in the lungs, hemoglobin stays almost fully saturated, around 98%, even if oxygen pressure dips a bit. That's a safety margin. The steep middle section means that at the tissues where pressure is lower, a small drop unloads a large amount of oxygen right where it's needed, and the curve can shift. A rightward shift caused by more carbon dioxide, more acid, higher temperature, or more BBG makes hemoglobin release oxygen more readily. That's the Bohr effect, and it's elegant. The most active, hardest working tissues automatically get the most oxygen. Finally, carbon dioxide and the control of breathing. Carbon dioxide is carried three ways. About 70% travels as bicarbonate ions dissolved in plasma, formed when CO2 reacts with water inside the red cells. That's the chloride shift. Around 23% binds directly to hemoglobin as carbaminohemoglobin, and just 7% dissolves in plasma as free gas. As for control, it's automatic. Respiratory centers in your brainstem set the rhythm, and chemoreceptors fine-tune it. Central chemoreceptors in the medulla sense the pH of your spinal fluid, which tracks blood carbon dioxide. That's the main drive. Peripheral chemoreceptors in the aorta and carotid arteries back them up and respond to severe drops in oxygen. Here's the counterintuitive part. It's rising carbon dioxide, not falling oxygen. That normally makes you breathe, and that brings our module full circle. Thanks everyone. If you like this, make sure to subscribe to find out more about physiology and other topics coming soon. Thanks, and have a good day.